So hello everyone. So on behalf of Indian National Trust for Art and Cultural Heritage and the INTACH Conservation Institute, I extend a very warm welcome to our speaker, Flavia Bartoli, and everyone who has joined us for today's talk in the Conservation Insights 2020 lecture series. I'm Dr. Padma Rohila, Director ICI Delhi. To introduce our speaker for today, Flavia Bartoli graduated with honors from Rome University in biology. Later on, she got a PhD degree in biology and a postdoc in biology applied to conservation of cultural heritage. She worked on several national and international projects for the conservation, like the conservation of Pompeii and Arcolano, Crypt of Original Sin in Matera, Lodge of Cupid and Psyche in Rome, and the famous Angkor Wat temple in Cambodia, and a Cappadocian site. She also was a visiting teacher for ACRA Foundation project in Zanzibar Built Heritage Job Creation and professor for the International Master Bioheritage Biology for the Knowledge and Conservation of Cultural Heritage. The topic for today's talk is Biology and Cultural Heritage, an introduction of positive and negative aspects. The cultural heritage, whether organic or inorganic, is subject to alteration and deterioration over time, linked to physical, chemical, or biological processes. The colonization and development of biological communities, the biodetergens, the biodeteriogens and organisms can cause aesthetical, physical, or chemical damages. It's one of the most important problems of conservation of all organic artwork, but also of external monuments, the biodeterioration. At the same time, the presence of some organisms can be favorable for the conservation of our artworks. The formation of patinas can protect them from alteration and chemical physical encoding of the environment, the so-called bioprotection. Finally, in the latest research, it has been discovered that there are some organisms that can be exploited for conservation intervention, and these are the bioremediation. This is the bioremediation. So today she'll be explaining all the various aspects of bioremediation, bioprotection, and biodeterioration. And before I invite Flavia, may I please request all of you to please put your microphones on mute. We will be taking the questions at the end of the session. So please do type in your questions in the chat box. I'll be looking out for those. And also type in your name, organization, and your email ID in the chat box. Thank you. Flavia, over to you.
Okay. Thank you very much. Good afternoon, everybody. I want to speak about the biology in the cultural heritage and do an introduction of the general aspect of the microorganisms and macroorganisms that can grow on our cultural heritage in different contexts, in different environments, and then in the specifically situation of the tropical environment, that it is the context where the organisms are in a very optimal condition for their growth. So the cultural heritage, the monument and the ruins exposed to the tropical environment are most deteriorated, are most subject to the biodeterioration due to the growth of the microorganisms. At the beginning, it is important, defined, but it is no well known at all that in any monument, it is suitable for the microbial colonization. Because the organisms can be can be autotrophic and heterotrophic. So it is indifferent for us if the subset it is organic or inorganic. So each cultural heritage done of wood, of stone, of paper, of tissue, of every type of our cultural heritage can be colonized by organisms. But the colonization of organisms, it is related to the physical characteristics of the substrate, but also by the climatic condition of the environment, okay? Because each organism have different ecological feature, and having different ecological feature can be suitable to colonize a substrate in determinate environmental condition, but maybe cannot be able to colonize in other climate condition, okay? Each environment, it is characterized by climatic characteristic, by climatic conditions that affect the suitability or not of the colonization. The term refers to its first definition by Hooke, and it is any undesirable change in the properties of a given material caused by the activity of living organisms. So the degree of change can vary greatly from disintegration and irreversible transformation of the substrate. So it is important to understand that the biodeterioration, it is a complex process that involves different type of process. Biodeterioration, it is different by biodegradation, because biodegradation, it is an irreversible transformation mediated by organisms of any material in natural environment, okay? Biodeterioration, it is an irreversible transformation mediated by organisms of any material, organic or inorganic, of material that have economical, historic, and artistic value, okay? This difference, it is important for the use of the terms, the correct use of the terms, and also for the correct analysis of the situation. In the major situation, the organisms responsible of biodeterioration and of biodegradation are decomposers. Decomposers can be responsible, are responsible of the deterioration of the substrate where they grow, okay? So in this case, we can see the difference between biodegradation and biodeterioration. Then it is important to define the term biofouling. Why? Because now in the context of cultural heritage, there is a little bit confusion of the use of biofouling, biopatina, biofilm, okay, colonization, patinas are all terms that refer to different situations of colonization. So biofouling, it is a term that means the accumulation of microorganisms, plant, algae, or animal on wetted surfaces, okay? In some cases, in the most cases of submerged surfaces, okay? In aerial environment, it is more correct to utilize patinas, biopatinas. So when we, for example, this situation, we have biopatinas, okay? That it is different respect to this situation that refers to a monument submerged in the sea, in the ocean, okay? Then it is important also define and stabilize the differences between biodeterioration and bioprotection, because the biodeterioration refers to the damage related to the presence of organisms. Then, instead, bioprotection refers to the positive effect that can be explained by the organisms that grow on the surface. And in the most cases, the biodeterioration refers to the process defined as umbrella effect, because the monument exposed in the outdoor environment are affected by different type of deterioration, whether in deterioration related to the environmental climate, so to the rain, to the wind, okay, to the erosion, refers to the weathering. Then also can be affected by the biodeterioration. So in relation to the environment where the monuments are exposed, you can have different effects by the organisms present on the surface. So the bioprotection refers to the balance between the presence of microorganisms and the presence of weathering agent. We can see an example. If I have a stone material, for example, because the major monuments and ruins present in outdoor condition are of different type of stone. Each country have a typical stone. In Rome, the marble and the travertine are the most spread. In Cambodia, the sandstone, it is the more spread. Okay, in relation to the country, you have different types, but you know very well this situation. So we have the stone, and the stone are exposed to different weathering agents. The weathering agents have an effect on the stone, on the deterioration of the stone. So the presence of the microorganisms, in this example, we have lichen or green microalgae, can, in some cases, can protect the stone surfaces by the weathering agent. But the real protect effect must be related to the biodeterioration effect of the same organisms. Why? Because the same organisms, for example, the same lichen or the same plant or the same biopatinas of microalgae, can have a positive effect in a country, but can have a negative effect in another country, because the growth of the organisms, it is related to the climatic condition. Okay, I can do an example in relation to the lichen. Okay, The lichen are spread in all the world, but the growth of the lichen can deteriorate the substrate. In the major cases, deteriorates the substrate because the biology of the lichen, it is to the presence of adhesion structure that permit to the lichen to live attached to the stone. Okay, so this adhesive structure penetrates inside the rock, and in some cases the lichen produce metabolic compound. Producing this metabolic compound and physically penetrate the substrate, this lichen biodeteriorate the stone, the monument where they grow. But their presence determine a maintenance of humidity on the substrate and reduce the change of the sun, rain, sun, rain. So the wet and dry cycle of the substrate, it is reduced by the presence of the lichen. So in some environments where the dry and wet cycle, it is very hard, the presence of this lichen can be sometimes considered as protective, because the biodeterioration apported by the lichen, it is less than the weathering, the deterioration due to the weathering agent, due to the dry and wet cycle. Okay, so the bioprotection, it is always related to the microorganism, the species of the microorganism or the organisms. I can do the same example for the Hydra helix, that is a climbing plant present in Italy and in Europe, okay, that in some country can have protective effect on the wall, but in other country it is very, 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 very dangerous for the ruins. Okay. So we can see the other terms, and we can see the terms of biodeterogen. The biodeterogen are the organisms that colonize the surfaces, okay, and that do a damage on the substrate. For example, this is biopatina of a green algae. Also, if it is red, but it is a green algae. And the species is Trentepolia umbrina, that form these big red biopatinas. Okay, this species, it is recognized as a biodeterogen for different motive. At first, from an aesthetic point of view. Why? Because the red color changed the color of the substrate, so the public, the human public, see this change of color as a biodeterioration. So the biodeterioration, it is related to 3 different processes: physical biodeterioration, chemical biodeterioration, and aesthetical biodeterioration. These 3 processes are in the complex biodeterioration process. And you don't have never only one of these three. These three processes, physical, chemical, and aesthetical, are always together. In fact, for example, Trentepolia have an aesthetical biodeterioration. He is an aesthetical biodeteriogen, but these species can also penetrate for some micron inside of the substrate. So also if it's damage, it is really, its physical damage, it is really low, this physical deterioration can be lead to a new colonization. For example, you can see that in this biopatina, you see red biopatina and green biopatina. The green biopatina refers to a lichen, powder lichen, Lepraria, of the genus Lepraria, okay? These lichen have two components. A lichen, it is a symbiosis of fungus and algae or cyanobacteria, in this case algae. So the fungus partner of the lichen penetrates a lot inside of the substrate. So the presence of the Trentepolia and the minimal deterioration of the Trentepolia can lead to a more colonization by more complex organisms, as lichen or mosses, okay? You can see here, in the environmental situation, you don't have never a single colonized, okay? You have colonized formed by a lot of organisms. In this case, you have a lot of lichen. In this case, you have lichen, crostose lichen, but also mosses. Okay, so the biodeterioration, it is a complex process that involves multiple organisms together. Why the biodeterioration occur? Because, as said before, the organisms can use the substrate as it has a past, okay? So you can see the heterotrophic organisms use the substrate as home and to eat, okay? But the autotrophic organisms, so you can see in this case the plant, but also the previous example of lichens, of mosses, but also of algae, use the substrate only as a support. But using as a support, they must be attached to the substrate. And attaching to the substrate, they penetrate inside to the substrate. So, the microbial colonization of a substrate results from different factors that interact each other: composition, climate, and type of microorganisms. So you can see here three different situations where you have three different ecological conditions of the substrate So you have different colonization. We can see the first example. Seeing the first example, you can consider that the presence of water coming from the floor permits the colonization of algae and other microorganisms. In this case, you can see the structure. See the structure. If you see the relation to the form of the structure, you see different type of biofilm, of patina. The red patina comes in relation to the cover, okay? The green patina grow where the cover is less. Why? Because in relation to the presence or not of the cover, you have more or less hydric input, water input, okay? The red algae needs less water input than the lichen. So the algae can grow also under the cover. The lichen, needing more water, can grow where there is percolation. Okay? I'm sorry. See also in this case. See in this situation, we can see that where we have the crack, where we have the junction between two parts, you have micro niche and micro habitat where the water can storage and where the powder can storage. Okay, so these presence determine the colonization by, as in this case, of mosses. Okay, where you don't have porosity, when you don't have ruggedness, where you don't have storage possibility of water and of powder, you don't have colonization. This case, you can see that there is a wall, an hole. Inside the hole, a plant, the seeds of a plant can be growth. So it is important to understand that each substrate have a degree of bioreceptivity. What is the bioreceptivity? The bioreceptivity, it is the tendency, the ability and the tendency of a substrate to be colonized. And the bioreceptivity of a substrate, it is related to the ability, to the characteristics of the substrate. In relation to the characteristic of the substrate, the ability of the microorganisms to penetrate, to growth in that condition, determine the colonization and the bioreceptivity of a substrate. So you can see, for example, in this case, you have a crack. The presence of this crack permits to these organisms to penetrate inside, okay? Where you don't have crack, the organisms cannot penetrate. But in some cases, the organisms have the ability to produce chemical compounds that degrade the surface and that create crack. Okay? See you another example of colonization, subsequently of colonization. Okay? What is the resulting damage? When you have organisms that use as nutrition our substrate, you have a more consistent damage. Okay? When you have different type of animal that colonized, it is simple to think to the mouse. When a mouse come inside to an home, but if you think if a mouse come inside to a museum, to a storage of monuments of organic material, you have or a whole destruction of the material. When the microorganisms, instead, when the microorganisms use the substrate as a source of minerals, as a support for the growth, you have a damage generally less relevant. But also this depends on the organisms, because if we talk of microorganisms, this is real, okay? But if we think to trees that grow in tropical country, so for example the trematales that grow in Cambodia, I have this example. If you, I don't know if you know, somebody know the Taprom temple of Angkor Wat, Cambodian UNESCO site. The presence of trees, it is a real problem. But these trees use only as a support. So the degree of damage, it is always related to the organisms that colonize, okay? We can see different situations where you have organic type of substrate, paper, and you have destruction by heterotrophic organisms. Also in this case, you have tissue, paper, and in this case you have fungi colonization. You can see the differences between animals that destroy all and fungi. Okay, it's real that if we don't intervene, in this case we have complete destruction. But at the same time of colonization, you have less destruction by the fungi respect to the animals, okay? At the same time of exposition. In this case, you can see wood artifacts. Okay, in the case of inorganic stone, inorganic material, such as stone, you have a different situation. How the biofilm form on the substrate, how the organisms can colonize this substrate. When you have an inorganic substrate, you have a process that was defined as ecological suggestion. So at first step, you have, in the major case, you have algae and cyanobacteria or bacteria, okay, that growth, that are autosufficient because do photosynthesis, so not need by nutrients from the substrate. Use the substrate only as a support. But with their metabolic activity and with their physical activity of migration, they can a little bit alterate the substrate. This alteration of the substrate determine a growth of ruggedness and porosity. This growth of ruggedness and porosity determine a more apport of water. The more apport of water determine the coming of more complex organisms that are lichen, for example, or mosses, or hepatic plant species, okay? But in the same time, the presence of the pioneer microorganisms determine the presence of organic matter on the substrate. So, heterotrophic organisms can come on the surfaces. And the ecological suggestion involve to arrive to a complex communities, okay? This is a scheme that explains what I say now. You can see in the time the evolution of biopatina from 1984 to 1996. You can see the differences of colonization. And from this type of colonization, you can, you can understand the water support, the water apport on the surface. And it is important to see that also after the restoration in the 1998, and also with the periodical maintenance, the colonization can be controlled. But in the same time, you can see that the colonization newly come, okay? Such as said before, the importance of abiotic factors in microbial ecology, okay? I said before that the climate and the environmental factor are very, very important. But in the same time, we said also that the surface bioreceptivity, it is important. But it is not negligible also the presence of pollutants, because the presence of pollutants determine the deposition of substance on the substrate that change the chemic of the substrate, so can determine the colonization. In some cases can also inhibit the colonization, but depend on the substance, okay? in the same time, also the treatments can be influence the colonization, because the type of treatments can apport substance to the surfaces, and so can function by a source of nutrients, for example, or can be inhibit if the treatment have a good effectiveness, okay? Have a good efficiency. In the same time, the ruggedness, Sam said before, the ruggedness and the porosity and the chemical composition are very important to determine the colonization by the microorganisms. Okay, you can see the different type of substrate, different evolution of the colonization. The same here, okay? Such as said before, our monuments and our ruins or our artifacts are ecosystem. You don't have a single organism, a single species, but you have community that grow in such environment. When you have community of organisms that divided the environment, that convive in the same environment, you have an ecosystem. So you can have macro ecosystem and micro ecosystem. In our cases of the cultural heritage, you have a micro ecosystem, okay? So you can see by this image that our statue, it is a real ecosystem with environmental characteristics that refers to the substrate, chemic and physical characteristic of the substrate, but also the sun, the rain, and the other environmental agent, okay? These abiotic factors are related to the biotic factor and to the communities of the organisms that colonize our surface, okay? So when the biodeterioration occurs, when? As said before, main environmental factor and materials variable we know well. Main environmental factor, water, temperature, and light are the fundamental environmental factors that determine the colonization, okay? But in the same time, also the variable of materials. But the water apport, I said a lot of time, the water apport, it is the most limiting factor that influence the colonization. The water and also, but after, the temperature and the lighting. The temperature and the lighting influence the colonization, but without water you cannot have colonization, okay? You can see here, in the same light condition, in the same temperature, nature condition, you have different colonization degree. Okay, you have different colonization degree. In this case, so you can see the different exposition to the general sunlight determine different type of colonization. So here you have no colonization, yes colonization, and it is related only to the water support. Here you have white colonization, black colonization. So the exposition to the light, but also a little bit to the water, you have different type of colonization. The ecology of organisms, of biodeteriogen, it is fundamental to understand if a colonization can or not, if a colonization can be affect your cultural heritage, your artifact, your monument, your I don't know, your ruins. It is important, very important, know the ecology of the organisms, because I can remove this colonization, but in the time this colonization come back after my treatments. Okay, same cases here. You can see the presence of water, the absence of water. In this case, you see the percolation event of the water. So you have a lot of colonization here, but not here. Similar here. You can see that this cover determine no apport of water from here to here, but after, where the water arrive, you have colonization. Okay, in the same time, the black patinas presence on the river Tiber, it is not smoke, it is organisms, cyanobacteria. Okay, cyanobacteria are autotrophic organisms, and they can grow on the substrate, on travertine, because the Tiber river are of organisms, are travertine, and you can see that the different apport of water determine different colonization. So we can see also that the different characteristics of the substrate determine different apport of water, and determine different apport of water determine different colonization pattern, different colonization spread. You can see here, or here in this example, that you have colonization only in some part of your wall. Why? Because these parts are related to a more porosity or a more ruggedness that determine a more apport of water, permitting the colonization by the mosses. Okay? In the same case of the Tiber River, you can see that some block of travertine haven't colonization. Why? It is related to the characteristic of the substrate, the characteristic of the material. Okay? You can see also that the organisms that colonize the substrates can colonize different parts of your surfaces. Okay? Different parts of your surfaces. And these different parts of the surfaces can be on surface, inside of surface, penetrating the surface. So you can have Endolytic organisms. Endolytic organisms are the organisms that colonize the fracture. Endolytic organisms are organisms that colonize the inside of your substrate. Epiphytic are only in the surface of your stone, and hippolytic are underground. Okay? So you can see, for example, in this case, that on the surface you don't have problem. See here, not visible problem, but you have problem of flake. Problem of flakes, it is related to the presence of organisms under the surfaces. Okay? They grow under the surfaces. In this case, the organisms colonize the part where the flake come off. Take off the flakes and you have colonization. Also in this case. So, who are the responsible of biodeterioration? Human can be birds, algal vegetation, ruderal plant, mosses and liveworm, lichens, algae, and herbal fungi, bacteria. All organisms can be biodeteriogen. Okay? They are example of microorganisms, microbiodeterogen, algae, and cyanobacteria, okay? And they can apply, as said before, different mechanisms, aesthetical, chemical, and physical damage to your substrate, okay? And you can have different biological agents, photoautotrophs, chemioautotrophs, and heteroautotrophs. Can see different examples of mechanical damage, such as said before, the presence of higher plant determine, this is the example of Angkor Wat. This species of trees, it is Trematelles that grow on the structure and with the roots destroy all. In this case, you have climbing plants. In this case, you are in a hypogean situation, for example, Etruscan tomb, okay, or Roman domus has Domus Aurea in Rome. You have the presence of algal plants on the surface, but the roots penetrating the soil arrive inside to your monument, okay? In this case, you have fungus that grow on your mural painting, and you have mechanical damage. this is an example of endolytic organisms that grow inside and determine detachment of your substrate, of your monument and ruins, okay, of your surface. The chemical damage, it is more difficult to understand, but it is related to the production of metabolic compounds related to the metabolic activity of the organisms that living release the such as excrement, okay, toxic compound that must be left and must be released, and these products are a lot of time acids and determine chemical damage of your substrate, okay. You can see here this situation, it is related to a chemical release by the substance, by the organisms. In other cases, the presence of organisms on the surface can determine the mobilization of chemical element present inside to the substrate on the surfaces. Aesthetical damage, I explained before, and it is related to the pigments present in the organisms on the biodeteriogen. In relation to this, I want to explain that for me, but it is in my opinion, the biodeterioration, aesthetical biodeterioration, related only to the pigments, only to the color of the organisms, not always it is a bad, but it is in my opinion, because it is related to the perception of the human, of the public. So maybe a human, it is more sensible to the red, to the green, or to the black. Why must be see by all has a damage? Okay, yes, it is a damage because you don't see the original color of your manufact, of your artifact. So on the pigmented, on the painting wall, for example, on the frescoes, for example, if you have pigmented organisms, you have an aesthetical damage. But for example, in this case, where you have only a wall, maybe the color can be seen as an aesthetical improvement, but it is a subjective perception, okay? In fact, now in the last time, a lot of artists use biology to do artwork. For example, a very famous artist, South African artist, William Kendridge, did a very big artifact on the Tiber River using the black of the microorganisms that colonize the Tiber River embankment. Okay, so can be seen as a bioart. The use of pigments of the organisms a lot of time was defined as bioart. Okay. The deterioration can be direct or indirect in relation to the type of microorganisms. it is important explain, as said before, that the climate context in which the monument is located as well its environmental history, and the biogeographical characteristics of the sites are all significant additional factors in biodeterioration, okay? Such as defined before. Tropical climates, I think that you know very well the tropical climate, so I don't spend time to explain the tropical climate. But it is important to understand this one, that the typical tropical climate, it is the least favorable season, the least favorable context for the optimal growth of the biological component. In fact, in the tropical climate, we have the biggest biodiversity in the world. So the climate condition of tropical context are very, very, very favorable for the growth, biological growth. And the ecosystem in tropical environment related to a multi-layered forest. Okay, so you have a very, very biological complex situation to monitor. And in the same time, you have the tropical context have the problem of the environmental agent, because the monsoon season and the problem of the monsoon season and then the dry season determine a very big dry and wet cycle that have a lot of degradation influence on the stone of the monument. Okay? Okay. So, in conclusion, although it is difficult to summarize in few words the characteristics of biological attack in tropical climates, we can summarize what has been expounded above in the following points. Considerable development on stone and plaster of the more hygrophilic plant growth, such as mosses, livewort, and peritophyte. So, in the tropical context, it is a very, very, very big biological colonization, and the control of the biological colonization must be due in balance with the climatic condition and with the wet and dry cycle. In the tropical context, you can see the balance between biodeterioration and bioprotection. Okay, with this I conclude my lecture. I hope that all is clear, but if it is not too clear, can you write to my email when you want? I don't know if there are questions on the chat. Padma?
Yeah, I can hear you, yeah. Any questions?
Yeah.
So far, my only question, I have a question is, how do you decide between a presence of a biodeterrent, whether it's reached a stage when it's a biodeterrent or it's still in the stage when it can be a bioprotective thing? How do you decide once you reach a site?
Okay, it is not too easy to decide. It is a very complex question, very interesting question, and in each case you have to do a diagnostic protocol, and you have to analyze the presence of dead organisms in few words, you have to identify the organisms, study the biodeterioration degree related to the presence of the organisms, and you have to identify the weathering degree related to the environmental factor. Then you have to evaluate the balance between the presence of these organisms, so you can understand if the presence or the absence of that organisms can be good or not. If you want, I can send you different paper that I write, I with Anna Laura Casanova, that will speak in the next 13th, and where we analyze this problem of the balance between biodeterioration and biodegradation, because it is not so easy to define if it is good or not, the presence of some organisms, and you have to do a study, an appropriate diagnostic studies on the case. So for example, we applied this diagnostic protocol in Cambodia and also in Cappadocia. And we have, for example, different results. So if you want, if you are interested, I can send you our paper so you can read read the case studies in different environmental conditions, so you can understand the complexity for take the decision.
Okay, thank you. I think please do that. I'll really love to read a little bit more. So we have questions now. Sahitya, the question is, do you think biofilms also have the capacity to act as bioindicators of atmospheric metal pollution? This is on outdoor sculptures and monuments in our urban environment. Do you think biofilms can help us to act as bioindicators of pollution?
Yeah. In some cases, yes. In some cases, maybe because some type of organisms, for example, lichens, if we speak about aerial pollutants related to the machine or to the motorbike or to the industrial, okay, quindi aerial pollutant, you can have some organisms, bacteria, and in some cases, and lichen in other cases, that are able to accumulate pollutant.
Okay.
But the lichen that are bioindicator and bioaccumulator of pollutant are also sensible to this pollutant. So a lot of time die in relation to the presence of a high quantity of pollutant.
Okay.
Okay, but can be useful. I think that it is an interesting interesting tool to study this aspect.
Are you aware of any strains of bacteria which adapt to the polluting metals such as sulfur and iron?
I don't know. I don't treat bacteria, but I know that there are studies. If you search in the scientific paper, there are different studies about the utilizing the bacteria to remove pollutant. A lot of time bacterial to remove pollutants by the substrates. Differently respect to the lichens, the bacteria were applied on the surface to remove substance on the substrate. Okay, differently by the lichens that absorb air from the atmosphere and absorb a pollutant by the atmosphere. I don't know if I answered to the question.
Okay, thank you. Another question is, can we use lasers or UV rays to remove biodeterioration, to kill the fungi and the algae, for example?
Okay, to remove the organisms depends on the organisms, and there are physical and chemical protocols to follow for remove the biofilms, and a lot of time and now in this time there is a big part of the research to study green biocides to use in cultural heritage. So for use extract of extract of oil trees, extract of plants, to use chemical agent, but natural chemical agent, so to not have problem of healthy of the restorer, because the restorer that are the only people authorized to apply biocides on the site. Yeah, that I think that the restorer must be associated by a biologist to define a removal protocol.
Okay.
Decide, because it is important to identify the organisms, understand what type of organisms you have, so you can decide if it is sufficiently apply physical removal, or if it is necessary to apply a chemical removal by biocides. Okay.
Okay. The question, have we tried lasers and UV rays? Have you any experience of using lasers and UV rays to remove these biofilms?
Yeah.
Ultraviolet rays and lasers.
I'm sorry, I don't heard. I don't.
Lasers and ultraviolet rays, UV rays.
No, I don't know.
You haven't used that?
No.
Okay. The next question is, with respect to trees growing on monuments, especially stone, do you find that certain stone substrates are more conducive to growth, encourage more growth of certain trees? Is there a relation between the stone substrate and the kind of trees that grow on it?
It can be, but not too much. It can be in relation to the environment, because, for example, in Mediterranean, I can explain about the Mediterranean ambient, the Mediterranean environment, because it is what I know better. And in the Mediterranean countries, you have such species that was defined as rural species that grow better on stone substrates. But if the stone, it is a calcareous stone, it is better for them respect if it is a siliceous stone. But it depends on the species of the trees.
So there is
yeah.
Okay, does biopatina alter the mineral composition of stone on which it grows?
Yes, because there are different species of trees that are, for example, were defined as calcareous species. So, for example, Quercus ilex, that is an oak species, that it is a species of tree typical of the Mediterranean area, it is defined as a calcareous species. So if the soil, if the substrate, it is rich in calcium, these species of trees grow better. Okay?
Okay.
But you have to know the ecology of the species that you are studying.
Okay. What is the best treatment to remove trees from stone structures without damaging the monument?
I'm sorry, I don't hear that well.
How do you remove trees from stone structures without damaging the monument?
Okay. Also, it is an interesting it is a very complex argument, because always the ecology of the species, it is important, because at first, to don't have problem with the monument, you have to intervene fastly and when the tree is young. Okay? And the trees is young and the roots are small.
Yeah.
When and it is more important, understand that the species of tree can have different characteristics. Some tree species, it is sufficiently cut the stem and the tree die. But in some case, if you cut only the stem, the tree reborn. So in that case, it is important when you apply the cut of the stem, then inject biocides inside the stem. And if you have also roots, visible roots, such as the case of a Cambodian site that I show in the presentation, you inject the biocide inside of the roots. In that case, you have the death of the trees. But if you cut only, the trees reborn, and you have a most big problem of that trees. But if you want to have a clear intervention without damage the monument, you have to intervene early when the plant it is very young. In that case, you don't have problem. But if you have big trees that have a lot of care, you have to do the remove of the tree only if you can do also consolidant intervent. If you cannot do the consolidant intervent on the monument, my opinion, it is better that you don't remove the trees.
Yes.
Because the growth of the trees take together the monument. If the trees died and you cannot do the consolidant event, you have the collapse of the monument.
Yes, that's true.
Okay.
Yeah. Another question is, can you suggest a microorganism that can act as a good biofilm as well as not damage the structure?
Oh no, I cannot.
Okay.
I cannot. It is
Okay.
a complex. I cannot.
Okay.
Yeah.
Is there any method to find out underground microbial growth in stone objects? Like the one you showed, is there any method to find out whether there's an underground microbial growth in stones?
I'm sorry.
The microbial growth which is growing inside, not on the surface, but inside the stone.
Yes.
Is there
Yes.
any way of finding it out, of finding out that such a growth is happening?
Yeah, when the micro, I don't know if I understand well the question, but I think that the microorganisms that grow inside of the stone are can be lichen, bacteria, fungi, algae, and also cyanobacteria. These 5 type of organisms can grow as endolytic, so inside to the stone, and growing inside to the stone determine detachment of the superficial part of your surfaces. So you have detachment problem of biodeterioration. But I don't know if I answer.
They have asked it if you can find this problem. How do you detect that such a problem exists?
Ah, how to detect?
Yeah.
Okay. Yeah, you can you can detect uh, seeing on the surface detachment of the surface. I come back to the previous slide so you can see the photo. Yeah, these photos, you see the presentation?
Yes, I can see the
Okay.
question here.
Okay, if you see this is a problem of microalgae that grow inside to the surface and determine the detachment of this part. So when you see this situation on your surface, I want to check another one. Yeah, when you have this situation, you can think that you have problem of endolytic organisms. Okay, you see detachment of this typology on your surfaces. When you have this type of detachment, you can think that you have colonization inside. But you can detach only if you have just a detachment of flakes.
Okay.
Okay.
Yeah. Thank you. Other question is that most of the the chemicals used for cleaning, chemical removal, or the so-called biodegradants are chemicals which have high levels of nitrogen, potassium, NPK. So will it not enhance the growth of microorganisms? That's the question. If we use chemical removal, does it enhance the growth again of microorganisms?
Yeah, it is the problem when you apply a biocide. So when you do a mechanical or a chemical remove of the biofilm, in the time you have to do periodical maintenance because the organisms come back.
Yeah.
In the time organisms come back. If you want to apply a long term, an intervent to a long term, you have to do what was defined as preventive conservation. So the preventive conservation, it is different to the biocide, to the mechanical or chemical intervent. The preventive conservation refers to the study of the climatic condition of your site and study the organisms present in your site. So you studying the ecological features of your organisms, you can stabilize the microclimatic condition of your site in which you don't have colonization. So if you can change the microclimatic condition of your site, you are doing preventive conservation and you can have not colonization for long time. Different by the biocidal intervent to mechanical that have a long time conservation, but not so long as preventive conservation.
Yes.
So if you, okay.
Yeah. Now, other question is about the essential oils that you talked about. So, can you share the essential oils which have been used as prevention against growth of microorganisms, the green biocides that you were referring to?
Yeah, if you want, I can send the paper that it is a review of all the paper that try to use natural compound as biocides. I can send you, Padma, and you share with Utens.
Yeah, I'll do that.
Okay.
Is there any, are you aware of any literature on use of eucalyptus, rosemary, or clove extracts? They can be used as inhibitors. Are you aware of use of eucalyptus oil, rosemary oil as inhibitors?
Padma, I don't heard you. I'm sorry. Can you repeat?
Regarding the green biocides,
Yes.
is there any literature suggesting use of eucalyptus oil or rosemary oil or clove? You know, clove.
Yes, yes, yes.
Have they been used as inhibitors anywhere?
Yeah. I see a paper on clove oil that have a good have good efficiency against bacteria, but not so much on the fungus. But they are experimental work.
Okay.
Also for the eucalyptol have a good efficiency on the fungus. And the other one, I don't understand the third one.
Rosemary. It's very common in Europe.
Rosemary. Yes, yes. Also the rosemary, it is good, very good.
Okay. Now,
But Padma, are all experimental paper.
no problem.
So,
Yeah.
okay.
Yeah, I understand that. So,
Yeah.
another question is, can thermal imaging be used to detect presence of insects or any organisms within an artifact? Can you detect the presence of insects or organisms within an artifact through thermal imaging?
Yeah, no, if you can see, you have this type of trace on the wooden when you see when you see detachment, you have proof of the presence of something. But I can suggest that if you know a biologist, you send the picture or you contact the biologist, because the biologists have can recognize more easily if it is present deteriogen agents or not. Okay. Also, yeah.
People are asking, can we share your email address with them if they want to contact you?
Yeah,
Can I
it is
share
here.
your,
Yes, yes.
yeah, can I?
Absolutely. Yeah, it is here. You can see?
Yeah.
Okay.
So everyone,
if you want, Padma, you can share my email address.
I'll do that. And also, if you can send the papers to me, I'll send it.
Yes, absolutely, absolutely.
Any other questions? I think there are no more questions. Thank you so much.
Okay.
I understand yours is a busy schedule. And thank you everyone for being patient and waiting for us and participating in this talk. We need your support. Thank you so much. So thank you.
Thank you so much. Thank you so much to you for your time. I'm very, 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 very sorry for my late. And
It's fine. I also take this opportunity. I think I never do it. So also thank people who have helped me with the registration, Ashwarya, Merin, Sushant, who helps us in uploading these videos. So the video will be uploaded. I've recorded this session. The video will be uploaded. I'll give you the link. I'll give the link to you also, Flavia. So if
okay.
anyone wants to see this session, they can visit our YouTube channel. The video will be there. So
Okay.
one.
Thank you very much.
Yeah. So next talk is by Anna Casanova on analytical a diagnostic tool for determination of biodeterioration.
Yes.
So I think some of the questions will be answered on that day as well.
Yes.
So
Yes.
please join us
Yes.
then, everyone.
Yeah.
Bye.